Cells are incredibly complex, and to help unlock the inner workings and the mystery of life and how cells go awry in diseases such as diabetes and cancer, we need to understand and see cells at very high resolution. In the last few years, there's been this wave of new advances in these super resolution microscopes where now we can see cells at the nanoscale. However, there's a really big problem, and that is typically we can only see these cells for just a few seconds before the dye photobleaches away.
We realized that two problems had to be solved. First, we had to figure out a way to label the dye very densely, and second, we needed to figure out a way to keep the dye from self-destructing. And the trick is to link the dye to a lipid, the greasy molecule that forms the boundary around almost every organelle in the cell. Linking the dye to the lipid does two things simultaneously. First, it keeps the dye in a state in which it doesn't absorb light, and second, it increases the labeling density by about 2 orders of magnitude. And by combining those two properties, we're able to visualize movies for 50 times longer than what was possible previously.
For instance, we're really interested in the endoplasmic reticulum, or ER. This is where proteins are made and subsequently sorted. Now, using our HIDE probes, these high-density lipid probes which spontaneously blink, using these lipid probes, we can now see the ER for tens of minutes. We see these wonderful processes. We see these ER sheets. We see these reticulated lace-like ER. We can see how it changes and remodels, how these tubules form. And we can now go from 2D imaging into 3D. So we're getting these new wonderful insights into the secrets of life under timescales that we could hardly ever imagine before.
So what's really great is that the HIDE concept is general. In theory, all one really needs is a lipid that localizes to an organelle of interest. We're excited to apply this to study interactions between organelles, and we're especially excited to use this to study interactions between lipids, which is something that has been extremely difficult to study any other way.